right, that give you more information about our services that we all provide. In case like you forget the information I announced here. But essentially our hours are Monday through Thursday, 8 a.m. to 9 p.m. On Friday, it's 8 a.m. to 5 p.m. And on Sunday, it's 1 to 5 p.m. So what are the resources that we have? We have a computer lab with over 20 computers that you can use. You can also ask for headphones and you listen to any content, as well as you can copy and print for free. A lot of students definitely use that. It's very beneficial, definitely take advantage of any free resources. As well as we have a conference room that you can reserve. Let's say you have a study group that you'd like more of a private feel, or you're involved in a student organization and you need a place to meet on campus. We have that reservable to okc.tutoring@okstate.edu. That information is on the STEM Lab business cards, as well as the rack sheet has our website. So, another thing you could also reserve is the testing room. Let's say you have tests with Lockdown Browser. We have a room that already has Lockdown Browser installed, as well as it's make sure it complies with those said regulations for Lockdown Browser. Or maybe if you don't have a place at home or somewhere where you need to be essentially secluded to take an exam, we have that available on campus as well as you can reserve. For the writing center, that's more for like if you have any writing related assignments. That's basically the majority of your classes, having to do like reports or anything like that. Like I mentioned, they can help with any class. It doesn't have to be just a Comp 1 or Comp 2 class. It could be any class where you have a writing related assignment. Sorry, it's been a long day, so I'm trying to get my words all together. That's what I'm saying a lot. I know this information deep in my core. It's just a long day. So yes, we do have essay Dropbox service available. Let's say you can't make it into the writing center like in person or virtually. You can email the okc.tutoring@okstate.edu email. And essentially when you send that email, you want to make sure you send your assignment with the rubric guidelines, so you can get the adequate feedback. With the writing center, you can do walk-ins. However, I would recommend appointments, just so you can get that one-on-one.
So she can send
And like I mentioned, with the STEM lab, really great for this class. I have the business cards and the rack sheet. They give essentially tips specific for this class on how to study, or really making sure you're understanding the concepts. It is walk-in only, so it's kind of first come, first serve in that sense for that center. Now, for the writing center, I already mentioned. So back to academic success coaching. As I mentioned, I'm an academic success coach here. And what that entails is really making sure we help students navigate college, whether you're a first-time student and it's been a few years, or it's your first time ever being in college, whatever the circumstance might be, we can help you make sure that you know that resources are available, as well as I'm here to help you navigate that. So that includes how to note take, how to deal with test anxiety, how to handle time management, really anything that you'll face in college. We are happy to help in any way we can. And for academic success coaching, we have online and in-person sessions. You can do walk-in and available, of course, sometimes we have appointments, so we need to make sure we set up an appointment in case I have an appointment in front of me. But yeah, like I mentioned, we help with various things, but outside of that, the SOC, once again, is free. Services are free. All of our resources are free. Really beneficial. A lot of students go there and it definitely makes an impact on how they perform. So in order for you to be successful in classes, you have to advocate for yourself and doing the taking advantage of resources. Yeah. That's it. I was gonna say I do have the rack sheets as well as business cards of my own and the STEM lab. If you don't like them, I'm gonna go ahead and take them afterwards. So this is the time to kind of do it.
Anybody want to grab some information on SSOC? You can come up here now.
Also, if y'all have any questions, y'all can let me know. Now.
Let's see.
As well, if you have any questions about the SSOC and you got the fact sheet, you can email the okc.tutoring@okc.edu email address, or you can also email me or call me, not text me. I just have to make that easier. So yeah, you can call me and let me know, hey, I'd like to get more information, or I'd like to walk through the SSOC and see kind of how that looks. Anything you'll need, I can help. So yeah, other than that, does anybody else have any questions? Does anybody else want to action, anything like that? No? Okay. Well, thank you guys so much. Y'all have a great rest of your day. Thank you.
Thanks for coming out.
Thank you for having me. Oh, Dr. Desiree is sick. So she won't be here.
Okay. Thanks, you too. All right, is everybody ready to get started? So they're logged into Kahoot. We're going to go ahead and get started with the Kahoot. It's going to be reviewing what we learned in chapter 4. I'm going to turn sound on so it's not quite so like ominous in here. It is really quiet. I hate the quiet. All right, since bacteria don't have a nucleus, they can't have DNA. Is that true or false? All right, so this is false. They have a nucleus. Sorry, they don't have a nucleus, but they still have genetic information encoded by DNA. So we, as eukaryotes, the main difference there is it has a true nucleus. That just means we have that membrane-bound nucleus. They still have all of the same functions of a lot of the eukaryotic cell functions, but they just aren't bound by membranes. And so a lot of the times when they have things that they have to do that occur on a membrane, such as making energy or photosynthesis and things like that, which we'll learn more about in chapter 10, but those things will happen on their cytoplasmic membrane instead of any other structures, and everything's just kind of floating around in the cytoplasm. So. All right, next question. Peptidoglycan layer is what in gram-positive bacteria compared to gram-negative bacteria? For gram-positives, it would be which? Right, substantially thicker. So our gram-positive bacteria have a thicker cell wall, and the gram-negatives, they have that thin peptidoglycan layer with that outer membrane around it, right? And so that's how they compensate for that. We're only on, this is only question 2, William, so that you know you're not that far behind. Got plenty of time to join in. All right, question 3, the gram-negative outer membrane contains what? This is another one that's kind of tripping people up today. All right, you guys did pretty well with this. Most of my other sections are choosing exotoxins. You guys did great. You didn't really choose that one. The answer is endotoxin. These are toxins that are actually part of the gram-negative outer membrane, whereas exotoxins will be actively secreted by bacterial cells. You can be gram-positive or gram-negative to do that. Lysozyme is an enzyme we'll talk about later on in the course. It breaks down, it's made by our cells to break down peptidoglycan, actually. And then lipoteichoic acid and teichoic acid, they're not really toxins, they're molecules that help support that thick peptidoglycan layer of the gram-positives. All right, what is acid-fast staining? So while you guys are answering this, Next, on Thursday, this Thursday, we will have another visitor or two. I think we have two coming. They're coming just to observe me teaching. It's just part of making sure that our professors are teaching what they say they're going to be teaching, and observing how I'll be teaching the class. So we will have two strangers, if you will, sitting amongst you guys, taking notes on me. So if you guys see that, it's just going to be business as usual for us. We're not doing anything different, but just FYI, that's why they're there. Anyway, so acid-fast staining, what this is staining to determine the presence of mycolic acid. So this is that waxy stuff that is on the surface of mycobacterium. And so the next question is gonna deal with our other ones that we often confuse the two. So we will have, this is mycobacterium species. And as scientists, when we are talking about bacteria that are called mycobacterium something, like Mycobacterium tuberculosis or Mycobacterium leprae, that's their name, we would write it as Mycobacterium spp, species. And so we were also following binomial nomenclature, we would underline it. Just FYI, reminding you guys of that. The Mycobacterium species, these guys have mycolic acid, which is that wax. And we use a special lipid soluble stain in order to bind to that wax called carbofusion. We'll be doing an exercise 4, which will actually be on the same day as our exam next week. So yeah, an exam, by the way, next week. So exam 1 is on Thursday next week, just FYI, so that you guys are thinking that it is coming up. So if that urgency kicks you guys into study mode, it's coming, man. It's right around that corner. So next Thursday will be the Unit 1 exam. While I'm talking about that, Tuesday of next week will be our review day. So normally I won't hold in-person reviews. That's what you guys' presentations are for those Unit 2, 3, and 4. But Unit 1 will be me giving a presentation over 8 major topics from Unit 1 in styles that you guys could maybe use as inspiration when you're doing your own reviews. It's meant to be relaxed and casual. And when you guys give your presentations, I do expect you to cover the material that you would be expected to know from the study sheets. But as long as you're covering that, you can do it in whatever fun or interesting way that you'd like to do it. So I'm going to try to have a couple of some variety in there. We will have Kahoot quizzes. One of them will be like traditional, where I'll walk you through it. Maybe there'll be like a game or like group competition or something like that. I have handout for one of them. I might be doing a song for one of them. It's, like I said, not going to be great, but it will be fun. So there's that stuff that we have to look forward to. We'll still be doing our chapter 7, because we will be done with chapter 7. So we'll have a chapter 7 Kahoot for our normal attendance thing. We'll jump into that review stuff. And assuming that there will be time afterwards, and hopefully there will be, after I've done all of my 8 topics, we will do the 5 questions from the critical thinking questions that I posted on the announcements. So we can walk through those and see how they're supposed to work, assuming we have time. I think we will. Anyways, so that's that. And we can talk more about how to study for that when we get to that. Okay. All right. So next question. True or false? Mycoplasma is an acid-fast positive organism with a thick, waxy cell wall. All right, so this one is false. And so the reason this is false is that mycoplasma is not the same thing as mycobacterium. And this is how I'm gonna try to write it out and like show them next to each other. How I will know so seeing it visually will help. So mycoplasma, like Mycoplasma pneumoniae, which can cause walking pneumonia. For example, these guys, this is the one where I think of that cup O plasma, which if it is spelled, that's not being spelled still. So, and it will have no shape. These guys don't have a shape. Why? Because they don't have a cell wall. So they don't have any cell wall. Okay, so these guys still have that thick peptidoglycan, but they have waxes on the outside. These guys down here, the mycoplasmas, they're the ones got the cell wall. Okay, and so think of these as being just regular little bacteria. Regular little bacteria have a cell wall. And these are two deviants from our normal situations. Where we think of gram positive as being thick peptidoglycan wall, we think of gram negative, thin wall, extra membrane. These are two deviants from that. Okay, so we have the wall with the wax on it, that's a deviant, but it's on the gram positive side. And these guys just straight up don't have any cell wall. So these are on that gram negative side, the positive side, gram negative side, but they're deviants still, so they don't have the normal situations. So these are the two exceptions that we have on that cell wall aspect of things. They both are named myco something, so we got to have a way to remember which one's which. All righty. Question 6, what are bacilli? What is that term referring to? All right, as far as cell shapes go, we have two biggies we have to remember. Bacillus is single, bacilli is referring to multiple, and coccus, which is single, and cocci, which is multiple. So bacillus versus coccus. A bacillus is rod shaped. We remember that because the L's are rod shaped, and the coccus is round or spherical shaped. Remember that because the C's and the O's are round. Simple way to kind of remember that one. So we only have two cell shapes, but we have three kind of new terms for arrangements, which we're going to get to later. I think it's the last question. All right, what does peritrichous mean? Remember trick, talking about like hair. So what is peri talking about? All right, so peritrichous means that it's all over, distributed all over the cells. Monotrichous, it has one. Amphitrichous, we're at each end. And then the tufts is, I like, this means tufts apparently. It means we have the tuft at the end, that's going to be lophotrichous. And you can have bacteria that don't have flagella as well. They'd be non-motile if that's the case. All right, number 8, the bacterial capsule. Which of the following would fall in line with what a bacterial capsule does? All right, so our bacterial capsule is that sticky coating that makes it harder for our white blood cells to eat up the bad guys. That process where they eat up the bad guys is phagocytosis. Phago means eating, and cyto means cell. And so it's hard for them to eat those guys up. This protects against phagocytosis. They're also useful for two other things, which would be adhesion and creating biofilms. All right, next thing. When are endospores produced? When would you want to produce endospores? All right, so endospores are produced in harsh conditions. So it's going to mean when we have a lack of nutrients or a lack of water or something like that, something where we want to hunker down and wait for better conditions. So that's going to be, in this case, our choice would be in nutrient deficient conditions. Again, believe it or not, you guys did better than the other sections. Like honestly, the other sections were almost all picking in moist, warm conditions. We'll make vegetative. cells in our warm, moist conditions. So those are active, multiplying. Most bacteria actually thrive pretty well in warm and moist conditions. So we don't need to go hunker down for that. So we're only going to be making endospores when things are going south, when things are bad. All right, how would you describe the strepto arrangement? I haven't written it on the board, but it's one of three kind of newer terms. There's two other terms like diplo or tetrad, which we kind of understand what those mean based on the words. Okay, so our three new terms are strepto, staphylo, and palisades. Staphylo is when we have our irregular bunches. Strepto is when we have chains or strips. I think of it as strips because it's easy for me to correlate strep with strip, but chains. And then palisades, these are like our fence post arrangements. And we have bacilli that are up next to each other, and only bacilli can do that. Cool. All right, here's our podium. Good job, everybody. You did good. All right, good job, guys. Okay, let's pull up our talk. So today we're going to be going over the eukaryotes. These are the guys that have true membrane-bound organelles, including a membrane-bound nucleus in them. So that's how they're basically defined as. And that is the biggest defining feature of those guys. And before we get into the talk, we have for question one, if we get to them, question one will be Jordan, W. Then we have Kelsey. Then we have me. Then we have Maddie, M. And then we have William. And so you guys, that'll be the questions that we'll be covering today if we get to them. You know the drill with that. So endosymbiotic organelles. We think that some of our organelles, we have very good evidence for this actually, we'll talk about it later on in the chapter, may have actually originated from like a progenitor cell eating up a bacteria. And then that bacteria that it ate up made like energy or something for that bigger cell. And that created a symbiotic relationship that lasted. And so there's a lot of evidence that will support that, which we'll go through in more detail later on. But that's introducing this concept of the endosymbiotic theory of where our eukaryotic cells originated from. Okay, so on the left side, we have the characteristics of eukaryotic cells that all eukaryotic cells have. All eukaryotic cells have a cytoplasmic membrane. It does the same thing as it does for the bacteria. It contains everything that's in the cytoplasm, all the organelles, everything. This is just that outer membrane that contains it all and also controls what goes in and out of the cell. We have the nucleus. This is a membrane-bound organelle that contains our genetic information. Then we have a few organelles that we'll go over in a little more detail that you've probably heard of in the past. Mitochondria, which will make our energy. The endoplasmic reticulum, the site of like large macromolecule synthesis, especially our proteins. The Golgi apparatus, which is basically our packaging and transport. And then vacuoles, which are for storage. The cytoskeleton isn't really a true organelle because it's not membrane bound. It's just literally like tubules and filaments of little proteins that act as highways that all of our cell activities are going to be happening along. They also keep our organelles in place. And then the glycocalyx, which is a term we learned about the bacteria, right? So glyco being sugar, calyx being a coating. And this is, could be in the case of unicellular eukaryotes. And yes, there are plenty of unicellular eukaryotes. Those can just be capsules and slime layers, just like they were for bacteria. But for our multicellular guys, they have a different function, okay? And some of our eukaryotes sometimes have a cell wall. Obviously, our cells don't have a cell wall, but plants do. Plants have a cell wall made of cellulose, and so do fungi and algae. And we have locomotor appendages. Instead of just saying flagella here, we don't just have flagella. We have cilia as well. So those are other appendages that we can use for moving our cells around or moving our environment around our cells. And then we have chloroplasts. These are used for photosynthesis, where things like plants and algae can make use of energy from sunlight to create their own cellular energy. All right, so here is a little diagram of our eukaryote cell. We don't need it too much for now, but it would be good for labeling where these things are in the cell. And then you guys could maybe go ahead and quiz yourself on what each of them does after we've gone through them. This is starting off with our locomotor appendages. Our flagella for our eukaryotes are a lot thicker. They're also membrane bound. Our cellular membrane is continuous around our flagella. And we actually have quite a few microtubules, which are our bigger versions from our cytoskeleton. Those guys create structure within it, and we can actually move it in a directed whip-like motion. So we can direct our flagella with these guys. So a lot more directed movement as a result of that. So bigger whipping motion as opposed to that rotating protein hair like we have with the bacteria. And we also have cilia. These are tiny, tiny little hair-like things. They look a lot like the fimbriae on the bacteria, but these guys move with like an oar-like whipping motion, so tiny. They will literally all work in tandem together to whip like fluid around them to help with feeding. They can also absorb nutrients from their environment. They give more surface area to absorb from. It's actually what the cilia on the insides of your intestine will use to absorb nutrients from their the digested food and matter that goes through your intestines and help move that stuff through along with the aid of your smooth muscle movement of your intestines. So our cells have cilia in our gut, but they're not using them for locomotion of the cells, but for locomotion of their environment, which is common for a lot of these eukaryotic and multicellular organisms. But we do have unicellular eukaryotes that just use these for moving around. So that can be used for moving the cell or, again, dealing with their environment. Moving on to the glycocalyx, like we said, that sugary coating. This is going to be, again, just a sugary polysaccharide coating. Maybe there's some proteins involved in there. And for multicellular organisms, this wouldn't be a capsule or a slime layer so much as it would be our extracellular matrix. So this is what would be holding our cells together to make our tissues and our organs. So it helps comprise that and delineate the difference between our tissues versus our organs in our multicellular makeup. So that's our extracellular matrix. And this would include things like hyaluronic acid or collagen and things like that can be incorporated in with all of that. And we have the cell wall. We only see this in our fungi and our algae and our plants, but we're not really talking about plants in this course, right? They're not microorganisms and they certainly aren't as important for causing disease. So our fungi and our algae, in our algae, they're always going to have cellulose walls. The fungi can have cellulose or chitin-based cell walls. Pretty interesting considering that's what makes up a lot of like insect, exoskeletons. Makes them crunchy. But it's just for rigidity and holding shape of the cells. When we talk about algae, they can be unicellular, so they can have single-celled cell wall algae. Whereas with plants, you can see how, and fungi, you can see how that helps with maintaining the structure of those organisms. All right, the cytoplasmic membrane, this is not news. It does the same thing. It just maintains its shape of the cells, especially for those that don't have walls. It also helps with maintaining that selective permeability, keeping what's inside and what's outside. Important for transporting nutrients and keeping nutrients in and all that. All right, moving on to our organelles. We have the nucleus first. This is the control center of the cell. This is the site of our genetic information, where we're going to store a majority of our genetic information. Within the nucleus, there is an area. Now, this nucleolus is not an organelle because it's not membrane bound, but it is a structure that we can see from staining within the nucleus where we have all this DNA. Well, there's this part of where our DNA is where we're making just a ton of ribosomal RNA. And we've already mentioned how your genes code for proteins or they code for genes that control expressing those proteins. And that's what your genes do. That's it. And so one of those genes that would help with making proteins would be the genes that code for ribosomes. And so when we build ribosomes, they're made up of ribosomal RNA, and we need to make a lot of them to make a lot of protein because pretty much everything in your cells is dictated by proteins. So that area where we have a lot of ribosomal RNA being made is called the nucleolus, just so we know. The nucleus itself, that membrane that it is contained by, that membrane is called the nuclear envelope. Alrighty, don't you love mitosis? So we're going to talk about how our cells kind of duplicate themselves as far as eukaryotes. In chapter 9, we'll talk a little bit about how bacteria duplicate themselves through a process called binary fission. It's not the same thing as mitosis. When bacteria divide, they have very much the same size daughter cells, like this all just the same size of everything. When we make daughter cells, our daughter cells usually are going to be smaller versions of the original cell, and then they'll grow once they have gone off to do their own thing. But this whole process is going to occur through these steps that we refer to collectively as mitosis. This is the duplication of cells. So we're making exact copies of cells. So when you are a baby, the whole thing that allows you to grow into an adult, making new cells and growing, this is mitosis. When you fall and you scrape your knee and you make new cells to repair that, mitosis. So anytime you're making copies of your cells that have the same exact genetic information, that's mitosis. When we need to make sex cells, we need to have a different chromosome number. As it turns out, humans have 2 pairs of each chromosome. I promise I should have just typed this. I can't type it with this. I don't know how to describe it to you guys. Anyways, we have 2 pairs of chromosomes. This is going to be one from the maternal side and one from the paternal side. Okay, and that you will have for your chromosome 1, you have one from maternal and one from paternal. For chromosome 2, you have one maternal, one paternal, and so on and so forth. And we have for each chromosome. So we have 23 pairs altogether. All right, so that's a total of 46 chromosomes. And so in order for new embryos to have only 46 chromosomes, the sex cells have to have only 23 from the egg and 23 from the sperm. Now that doesn't mean the egg contains 23 from your maternal. That isn't how that works, right? So when a mother, a female, who's becoming a mother from her eggs will all have a mix and match from her paternal and her maternal. And it's actually pretty interesting because what you can see is you can have maybe your chromosome 1 from your maternal and your chromosome 1 from your paternal. When you're making your eggs, those can actually cross over and splice across from one another so that your eggs have more genetic variability than just one egg has a maternal and one egg has your paternal, and that's what you get. So it's really interesting how that can happen. We're not going to go over that in this class. If you guys are interested in it, take genetics. That'll go in way more detail on that. Probably the only person who's going to have to take genetics in here, I think, is going to be Tommy. I think you might have to take genetics. Yeah. So, but it is really interesting and cool stuff if you're interested in that. But we have to separate out our cells and our chromosomes for making those gametes, right? Our eggs and our sperm. That's meiosis. And we're not going to go into how that works, but that's the general idea of what it is and why it exists for making our sex cells. So meiosis, these are for our gametes or our sex cells for sexual reproduction. All right. For mitosis, we're literally just going from a 46 chromosome cell to an exact 46 chromosome match, making copies, right? So there's 4 major steps. I know it shows a whole lot of steps in here, but you really just need to know 4 major steps. And that is abbreviated as PMAT. And if you have taken class with Dr. Shearer, and the way that he tells people to remember this is that if you get a new puppy, you want it to pee on the mat, not on your floor, not on, your belongings, but on the little pee mat that you set up for it. And that's how I remember it too. And when my dog got sick this morning, she did not pee. It was vomit and other things. Okay, we'll just leave it at that to save you guys from the details. But it was a lot of cleanup, and couldn't get all of it done in time to get to work, so I had to go back. It was a good time. It's how we love to start our day, isn't it? So, and she's fine. For those of you that are wondering, once she got it out of our system, she was like just fine. You know how dogs are. Like it's just magic. Like she had this horrible thing for just a minute, and then she's like, "Yeah, more food, please. Like I'd love to eat food and go play outside." And she's like 13. So you never know. You never know what's going to happen. So anyways, our 4 steps, right? So P represents our prep phase. This is called the pro phase. This is supposed to say pro phase. I'm just going to put it as pro. And that's the step right up here. This is the pro phase. And here we're just prepping for getting ready to divide our cells. So what that means is we are taking all of our copied DNA. So like I said, you usually have 23 pairs of chromosomes that exist like this, your maternal and your paternal. Well, during the process of getting ready for mitosis, you're going to make copies of your maternal and copies of your paternal, okay? And these are going to be joined in the center by a little protein setup called the centromere. and they're going to join them like that. So now suddenly this looks probably more like what you're used to seeing and what we see even in these pictures, which is our X's. So all of our chromosomes existing as X's. These are literally copied versions of your chromosomes. Normally, in normal functioning cells that are not duplicating, you have one line, not X's, just one line. You only see the X's form when we've made a duplicate of that chromosome. So the duplicate from your maternal side and the duplicate from the paternal side, because we're getting ready to rip them apart so that our new cells have one copy of the maternal on one cell and one copy of maternal in the other cell. And everybody has 46 at the end. Make sense? So the reason that we always see them as X's is because it's way easier to literally see them when we stain your cells, when we have the doubles made and we've condensed them all inside, because our nucleus contains a whole lot of a mess of DNA. And it's just spread out all over our nucleus. And so in order to get ready to split up our cells, we need to consolidate that down. And when it's consolidated down so it's not a big mess everywhere, because we have to get rid of our nuclear membrane in order to do this, we form it into a structure called chromatin. And this is just consolidated version of our chromosomes. It's all shrunk down and consolidated. So instead of being a big old mess like we see here, big old tangled mess, now we have consolidated each of our chromosomes down and they've been duplicated and all of that. We're also creating structures that are going to help with pulling them apart. I don't care what, we don't even worry about what they're called, okay? But that is all happening during prophase. This is our prep phase. Then our nuclear membrane is going to dissolve and we're going to line everything up in the middle. And this is in the metaphase. In metaphase, this is where we line up our chromosomes in the middle. In the middle. Okay, so this is our metaphase here. This is a good representation of just typical metaphase, where all those X's are just lined up right down the middle. Because what are we fixing to do? Rip the X's apart. The metaphase, lining up in the middle. In anaphase, we are ripping them apart. So guess what A stands for? Apart. In my mind, that's how I remember it. Anaphase. So here we go. Here's a pretty good picture of anaphase right here. Metaphase here and prophase there. And so we're just literally pulling those X's apart so we can start consolidating them on the different sides of what's going to become two cells. Then in telophase, I think of this as meaning T for totally done. I'll just put total because I don't want to write out the whole thing, but totally done. Once we're totally done, that is going to be when we're finally getting to that point where we're going to be able to recreate our nuclear membrane and start pinching off our membrane around everything and divide up our new daughter cells. Make sense? Easy as, what did I say, clear as mud? Yeah. All right, so I hope that that kind of helped a little bit out with understanding the 4 basic steps of mitosis, because there's a lot more involved in it than that, but that's the basics. Any questions about mitosis before we move on? All right, endoplasmic reticulum, or ER. I won't make you write it out, but you will have to like recognize these terms on exams, right? So the endoplasmic reticulum. We have 2 different versions of it, the rough ER and the smooth ER. The rough ER looks rough. That's literally why we call it that. Is we look at it in a microscope, it looks rough. It looks like it's like got all these like dots all over it. And those dots that are on it are ribosomes. And ribosomes are the structures that we use for making proteins. All right, they're going to literally, you'll have in your Smithsonian institution of your nucleus, have everything coded that you could possibly code for in a whole body. You just want to make keratin to make some hair or some nails or something, right? You just want to make keratin. So you go in there and you just copy down the recipe for keratin. That's going to be your messenger RNA. You can take that out of the nucleus and into the ER, and the ribosomes that stud the membrane of the rough ER will then read that written down messenger, that working copy of that gene, and spit out the protein as a result. Okay, and we'll talk about the literal mechanisms of how that works in chapter 6 in the genetics chapter. But for now, that's the basics of how that works and why there's ribosomes on the rough ER, because they're making the protein. They're a protein making machines. All right. In the smooth ER, that's where we're going to be making other macromolecules like our lipids and our carbohydrates and things like that that we need for storing energy, and building other aspects of the cell. So that all happens in the smooth ER. So this is our manufacturing plant. Oh my gosh, man. Back. If I stare at it really close, I am better at writing at it apparently. So manufacturing plant is the ER. This is kind of what it looks like. So they've done our little cartoon rendition of it. These little dots, the blue dots that are on here represent ribosomes, and they literally will clamp on to whatever that message is, and they can just crank out protein into that endoplasmic reticulum inside. So that's how that'll work. You can see here the reason we can actually see these membrane folds back and forth on each other is because they're studded with those ribosomes. Look at how big that endoplasmic reticulum is. This thing here off to the side, this whole structure inside of this red line is the nucleus. And so the endoplasmic reticulum is always going to be right outside the nucleus because you need to take your message out of there and then just immediately start making protein from it. So it's right there, ready for us to start making our product. Okay. And the next step is the Golgi apparatus. This guy is where we are going to have packaging and transport. So this is basically like your packaging, like the Amazon shipping facilities. Okay, they're like putting all the stuff you're ordering into the boxes and packaging up, making sure they're getting sent to the right place. So this is packaging and shipping. They can do some very like end stage modifications as well. So all those lipids that you may have made in your smoothie they can start attaching them to proteins if we need to, things like that. So doing our end stage modifications, our packing, and our shipping. That all happens in the Golgi apparatus. These guys look like a more globular version of the ER for what it's worth, but that's basically it. These guys are packaging and shipping structures. All right, they also create vesicles. And anytime you hear zome on the end of something, is what we talk about a lot, lysosomes and phagosomes and phagolysosomes. And zome just means vesicle. So anytime something's happening in there, zome is a vesicle where those things are happening. So the whole assembly line of everything that goes on in the cell is going to be message from the nucleus, make a copy, take it to the endoplasmic reticulum, make your product, package it and ship it in the Golgi apparatus. And then from there, the vesicles that come from the Golgi apparatus can go anywhere in the cell or secrete things out of the cell, whatever you need your cell to do. Think of all those messages that our cells are sending to each other and making chemicals to alert other cells to things going on, or sending messages between nerve cells, and all of these things that we could be doing. All of that stuff is going to be basically managed by products that are contained and packaged by our Golgi apparatus. And so that whole line is really important for sending messages like that. And this is just a picture depicting that with our nucleus. Our nucleolus is making our ribosomes to stud our ER. In the ER, making our product that's going to be transported to the Golgi, where it's going to finish everything off, package it, and transport wherever it needs to go in or out of the cell. That's that orange guy here, Golgi. Right. Next one, the vesicles. We have certain kinds of vesicles that have very distinct functions in the cell. One of those is the vacuole. The vacuole is a membrane-bound sac that is basically like our pantry or like our water storage. Okay, so that's really all that they mostly do. In plants, these are such huge structures that actually helps provide the rigidity of, whatever, like the flower. Like the whole reason that a flower stem can hold up that big old flower head is because those cells are filled up with vacuoles that have a lot of water stored in them. You don't water your plants for a while, they start to wilt, they can't support themselves as well because the vacuoles have been emptied. They've used up their stores of water and now can't use those as structural support either. So those sacks of whatever we're storing can be helpful in structure. Definitely the case with plants. The other vesicle that we want to be aware of is the lysosome. I've already briefly mentioned that lysozyme, which you won't be tested on lysozyme on this test, okay? But lysozyme is a protein that we make, an enzyme that we make that breaks down peptidoglycan. It's a defensive mechanism. And so we can pack these vesicles with lysozyme and other toxic things like free radicals, acid, and other things to help break down bad guys. And so we keep little pre-packaged vesicles of bad guy killing juices, and these are called lysosomes. And so when you go and phagocytose something with your phagocytes, your white blood cells will go pick up a bacteria and phagocytosis, eat it up into a vesicle that we would then call a phagosome. And we can merge our lysosome with our phagosome and make a phagolysosome. Yeah, don't you love it? So we'll have our phagolysosome and combine those chemicals from the lysosome with our bad guy and then degrade that. And that usually takes about 30 minutes to completely degrade down like a bacterium, for example. So when you have a whole lot of white cells out there doing that work, it can actually happen pretty quickly. So that is the function of a lysosome. We'll talk about that more in chapter 15 in our innate immune chapter. Now we're moving on to the mitochondria, and after mitochondria, the chloroplasts. These are energy generating organelles. These are membrane bound, but these guys are pretty unique because they look like this. They have an outer membrane, just like our other organelles do, but then they have an inner membrane, and their inner membrane actually like wraps back around on itself. I'm not doing it justice here. Hold on, I'm gonna draw it like this. It wraps back around itself. So sort of like your brain. So you know how they tell us that your brain has these folds to increase the surface area? Well, the inner membrane of these mitochondria has all these folds to increase the surface area. And on these folds, we have important proteins that are involved in aerobic respiration, which is how we get our ATP for our cells. It's absolutely the most important thing for making energy for the cells. And these are so important that, again, if I suffocate you, you die, because these guys are trying to crank out ATP to keep up with your energy needs for your cells. And we run out that fast, just in minutes. And so this is our structure in our cells, our organelles that make that energy. And they do as much as they can. You become a runner. Like you go from being someone who's relatively sedentary to somebody who does a lot of cardio activity. You're going to be needing more mitochondria in your leg muscles to make sure you're providing enough ATP for that. If you start doing a lot of activity, cardio activity, and your mitochondria can't get enough oxygen to make the ATP that you need to move your muscles, then what they'll do is they'll revert to basically what is the equivalent of fermentation, and they make lactic acid, just like bacteria do, in response to not having that oxygen. And so we get lactic acid buildup in our muscle, and that creates a lot of soreness and things like that. And that to you is a sign that, okay, well, our muscles are probably building up and regenerating. You'll probably get more muscle cells, but you'll also get a lot more mitochondria too. And so these are ways that our body will adapt to different activity that you might introduce to it. If you're doing it regularly, it signals to your body, we need to do more. So mitochondria are energy generators. These inner membranes and the folds that are in these inner membranes are called the cristae. I'm just put C here. And then the space that is in here is called the mitochondrial matrix. Inside the mitochondrial matrix, our mitochondria actually have their own genetic information that codes for the mitochondria. It is in a circle format. Ours, in case you weren't paying attention, are linear. Okay. What else has circular DNA? Bacteria, right? Interesting. They have their own DNA in a circle. They have their own ribosomes. Guess what their ribosomes look like? Bacteria ribosomes. Okay, they don't look like our ribosomes. They have their own ribosomes that look like bacteria, the same size as bacterial ribosomes. They make their own enzymes. They divide completely independently from our cells. So that's why your mitochondria in your leg muscles can start duplicating themselves. They don't need your whole cells to duplicate. This is not the case of any of the other organelles in your body. So not your endoplasmic reticulum, not your Golgi, none of that stuff. That's pre-programmed by whatever your cell is doing as part of your tissues. But your mitochondria, they can divide on their own. Their DNA, because mitochondria are kind of bigger organelles, you inherit your mitochondria from your mother. That comes with the eggs. Because the eggs are bigger than sperm can't carry mitochondria with them. They can carry the energy that they need for their journey, and that is it, okay? But eggs carry mitochondria. So every single human being on the planet got their mitochondria from their mother, and their mother got it from her mother, and so on and so forth. So every man also got their mitochondria from their mother. And so we have, maternal mitochondrial DNA that we can trace back throughout evolution and see changes in that. And we can basically use that to relate, relatedness or changes in the human evolutionary genetic lines and things like that. But anyways, yeah, they are completely like organisms in your cells, quite literally, and they align very well with bacteria. So this is our evidence that we have that supports that endosymbiotic theory, that in fact, we think that our mitochondria, chloroplasts are the same way. They have these same properties, that those two structures may have come from organisms that our cells ate up and then just became symbiotic with them. And it makes sense. They have everything there to support that. So really interesting stuff. So the chloroplasts, what do? We know they're for photosynthesis. They have these structures inside of them called thylakoids that are like folded membranes there that have all these pigments on them. The pigments get excited. The excited pigments allow the chloroplasts to make energy just a little bit. I want to be very clear about that. Just a little bit of energy, just enough that when they have accumulated energy from the sunlight, they can then use, regardless of sunlight, that energy to fix carbon dioxide into organic molecules. This is actually where plants get their organic molecules from, is from fixing carbon dioxide into glucose and then using glucose as their major stepping off point for their organic molecules. And so they create all of their organic molecules from scratch from carbon dioxide from the atmosphere. And this happens thanks to the energy they get from the chloroplasts. They do not get all of their energy from the chloroplasts. They just get enough to make glucose. So what's so important about glucose? Well, if you've learned about aerobic respiration, then you know what's important about glucose. It's our stepping off point for everything that goes off in our mitochondria, our energy making cells. So plants actually make just enough energy to make glucose. And then glucose is a huge, huge energy source for the mitochondria. I mean, you might only make a couple of ATP molecules from your cycle through photosynthesis, but for every single glucose molecule that you put through our mitochondria, you're going to get like 38 ATP from just one 6-carbon glucose molecule. And that is really impressive. So plants don't only go through photosynthesis. Plants also go through aerobic respiration. So they will also need oxygen in order to produce their energy, which is interesting because you guys probably don't think about that. We're like, they make oxygen though. Well, yeah, they make an excess of it while they're building their glucose, but they also need some of it. So they'll actually go through aerobic respiration as well to make their energy. That's a primary energy source for them. They just make their glucose. We just eat ours. So that's pretty much the cool part about plants. We'll talk more about that in chapter 10. Because plants aren't the only ones that go through photosynthesis, our bacteria do too, right? So those cyanobacteria, our photosynthetic bacteria, are probably the ones that built our oxygenated atmosphere for us way back in the day. So pretty interesting. Ribosomes are protein synthesizers, again, little machines that are going to make our protein. We have different sizes ones for our bacteria versus our eukaryotes. You don't need to know the sizes, but you just might need to know they're different, right? And that's part of the evidence for that endosymbiotic theory, supporting that maybe our mitochondria and our chloroplasts came from bacteria back in the day. So yeah, these are the structures that are going to be used to make our proteins. We'll get into more detail on ribosomes in chapter 6. All right, our cytoskeleton, again, are highways in our streets that allow us to move things around in the cell. They also keep our organelles where they're supposed to be in the cell, so they're not just sloshing around wildly. There's 3 different types of them, and I don't need you to distinguish between them, but just for the sake of understanding how the cell operates, the smallest ones are our actin filaments. Then we have the intermediate sized ones. Then the microtubules are our largest ones. Now we're going to move on to our groups of our eukaryotes. Okay, so we're going to start off with the fungi. We have fungi, we have the protista, and we have the helminths today. All right, so 3 biggies. So with the fungi, there's millions of species of them. We have the macroscopic ones, the ones we can see. They will have multicellular structures that come out, and we call them mushrooms. They'll have puffballs, gill fungi. And these macroscopic guys are usually just the fruiting body or the reproductive body of these organisms. And it's usually what's underneath the soil or wherever they're growing that is like what really constitutes their vegetative or like their main bodies of these. Okay. So what you're seeing as mushrooms, what's underneath, what looks like roots to you is not roots. Okay. It's their main body of the fungi. Then we have our microscopic fungi. These guys can exist as single cell organisms, but they usually work as colonies. All right, first we have yeast. Yeast are eukaryotes. Yeast are fungi, okay? So yeast, yes, I am talking about all yeasts, okay? They are fungi. They just are a certain structure of fungi. They look like this, right? They have their round cells, they kind of bud off one another with these a sort of random sized daughter cells that they will, build off, and those can break off and leave scars from where they broke off of the membranes and so on and so forth. But they bud off of each other and create these daughter cells that'll be sort of smaller in shape, and then those daughter cells can go on and grow up to be the same size as the parent cells and so on and so forth. So this is yeast. We're typically used to talking about fungi in terms of molds and mushrooms. Molds exist as hyphae. This is another form that they can exist at. And hyphae are those hair-like guys that sort of like grow out as hairs and branch off of each other. And we see it as like fuzzy or furry looking and stuff like that. So that's hyphae. You can have interwoven hyphae creating the entire mass of your mold. So if you look at like bread or strawberry or something that's molding, you can see that fuzziness on there. And that whole structure would be called the mycelium. But even mushroom or macroscopic fungi have mycelium. And that looks like roots to us under the soil. So those roots can actually be interconnected between those macroscopic fungi, our mushrooms and things like that. So we'll see mushrooms, and underneath all what looks like roots are actually the mycelium, or the actual hyphae, which is the vegetative body of these organisms. And there is an organism of fungi that's considered one single organism that's like over hundreds, if not thousands of acres of forest in Oregon that represents the largest organism on the entire planet because it's all interconnected mycelium. And so you might have individual mushrooms here and there, but it's all interconnected under the ground by these connected mycelium, the hyphae that are all connected under there. And that body altogether is called that mycelium. And the mushrooms that we see are just the reproducing bodies of that. Really cool stuff to think about. But so that's the difference between hyphae is the branching, the mycelium is that whole body, which for us on molds on your strawberry is easy enough to see the whole thing. It's a little more complex with our mushrooms. Some of these fungal organisms can switch between yeast type of organism and hyphae type organism. That's called dimorphic when they can switch. And our reproductive structures for fungi are called spores. Very important that you understand the difference between fungal spores and bacterial endospores, because bacterial endospores are not for reproduction, right? They're just for hiding out in harsh conditions. In bacteria, if you are a bacillus or a clostridium, you will make one endospore for one cell just to hunker down and wait out the conditions. These guys, we have this one mold creating all these spores that it can release for reproductive purposes. So these are like the opposite functions. These are only going to be made, these spores are going to be made in good conditions for the fungi. So fungal spores, reproduction. Not the same as endospores. And when I ask you questions on the exam about the spores in general, I will always call bacterial endospores endospores. Okay? I will not be confusing with that language. And if we're talking about fungal spores, I will specify that it is fungal spores. But just be aware that it's up to you to remember which one does which. So fungi can get their nutrition from several different sources. They usually get it from a wide variety of substrates, which means they are heterotrophic. So anytime you see troph, I always think of like a trough, like pigs will eat out of a trough and they'll just eat anything. So eating is what troph means. So hetero means different. So different things that they can eat, okay? And fungi can also be saprobes, which is a more specific kind of heterotroph, where they're literally just eating dead things. Or they can be parasites, where they're living off of living animals or plants. So when something is alive and they're eating off of it, they're a parasite. If it's dead and they're eating off of it, they are saprobes. Okay, so things that are decaying, they get fungi on it, saprobes. You get athlete's foot, that's a fungal infection, and that would be a parasite causing damage to the tissues of the host. All right, this is just an image kind of showing the ways that this mold can kind of reproduce and giving us a little precursor into what these reproductive bodies can look like at the microscopic level. We're going to talk about them in a little more detail. Okay, so how can fungi reproduce? A lot of ways, unfortunately. So first obvious one, just growing outward. You get a little bit of mold on your bread and you leave it there for a while, it's just going to grow bigger. Outward growth. Okay. Another thing that they can do is fragmentation. You get a piece of moldy bread, you break off that piece of moldy bread and rub it onto another piece of bread. it'll break off in pieces and it'll grow mold on that new piece of bread, right? So fragmentation, you can literally cut off pieces of the furriness and put it on there and it'll grow. So fragmentation, pretty easy. Primary mode of reproductive methods for fungi is going to be spore formation. And again, these are reproductive methods, not the same as bacterial endospores, which are non-reproductive. They can reproduce sexually. We are not going to be talking about that because it gets pretty complex, but they can reproduce sexually. But primarily, they're going to be doing asexual reproduction. And these are the two structures they can form. You either, as a fungus, make sporangiospores or conidiospores. Okay, so here's the difference. Sporangiospores are covered. They have like a covered sac around them and they'll burst out. Conidiospores are open and they'll just release them gradually. And those are the two main structures. And these can actually be pretty defining. I'm going to show some pictures of them in a moment. There's a pretty defining for a lot of fungal species. If you are a fungal species that does sporangio, then we can look microscopically at your fruiting bodies and see that cover on there. And that helps us know, okay, this is more likely to be this species than the guys that don't have the cover, the conidio kind, okay? So I don't have a good way for remembering these, unfortunately. I would share it. So like I said, identifying these guys, you can use looking if it has sporangio or conidio. You can look at the macroscopic, whether that's like the color of the hyphae, whether it makes hyphae or whether it's yeast-like, whether it's, has the macroscopic mushrooms involved and things like this, all for identification. So mushrooms typically are not going to be involved in disease, right? But the microscopic guys, the molds and the yeasts can be involved in disease. And we actually know that to be the case, especially if for women, you might be aware of yeast infections. It's actually pretty common to have yeast as part of your normal biome. When you take antibiotics and you kill off your normal bacterial biome, the yeast will overgrow. One of the analogies that I use often in my class is like a stadium. And normally in your microbiome, your stadium's filled up by a whole bunch of different things. And they fill up all the seats so the bad guys can't come sit down. You take antibiotics, you kill off all the bacteria in your stadium, and the yeast, who were just sporadic all over the place, now they have all these seats they can fill up, and they will. And that's what leads to a yeast infection. Same thing with BV, if you've heard of bacterial vaginosis, you're killing off your yeast and maybe certain kinds of bacteria. So now your whole normal setup of who takes up your stadium seats is thrown off. We have different guys taking up way more than they normally should, and that leads to bacterial vaginosis. So we have a lot of that going on in our body that can lead to that. C. diff is another great example of that. And we'll talk about those sorts of things a little bit later on as we talk about disease and all that. But for fungi, it's normal to have fungi as part of your biome, but in certain amounts, in certain areas. And when those things get out of control, that's what can lead to disease. This is a great picture to depict our sporangio spore forming guys and our conidio spore forming guys. So you see how these guys are closed, they're covered, and these guys are just open out to the air. Two good examples of open air conidio types would be aspergillus, which is a really common fungus that's in the air. It's like all over the place. And penicillium, which you've probably heard of. It's where penicillin comes from. It's actually super common in our environment as well. And for penicillium versus aspergillus, on a petri dish, they look very similar. They get a fuzzy greenish kind of growth to them. But you look at them under the microscope, they're both C's, they're both conidio, so they're both open. But the aspergillus has like a dandelion, big round dandelion shape to it. And then the penicillium looks like paintbrushes with little balls they'll break off. Very distinct from one another. We'll actually get to see them in our eukaryote lab when we get to that in the lab. So these can be used for identifying. We can have medical conditions associated with fungal infections. I know. We can have medical conditions associated with fungal infections. Most of the time, it'll be introduced from our environment, and most of the time, the fungi just doesn't normally cause disease in us. It just usually exists in our environment and is introduced in new ways. One good example might be if you get a sinus infection or if you have allergies. So you have allergies, let's say just to like pollen or something like that, or hay fever. You get irritation in your sinuses and the inflammation causes the tissue in your sinuses to swell up you can actually trap fungal spores within the swollen tissue in your sinuses, and they'll grow and be happy in that nice, warm, protected environment. They've got food around them. They got the bacteria and the mucus and the sugars from your mucus and all this stuff that they can feed off of. And if you can't get it out of your sinuses, they'll be happy to just grow there. And they can grow whole big mold balls up in your sinus cavities. And this is especially true if you are allergic to mold. So it's just adding on to the inflammation. And that inflammation can become so severe and so prolonged that that can become almost permanent tissue, leading to nasal polyps and things like that. And so that can require surgery to clear up those things. Medication can help if you catch it early enough. But so fungal sinus infections can happen as a result of that, but it's not normal for that to be happening. It's not like it's trying, that's how it exists. Like, COVID, that's what COVID does, is cause COVID. But the mold is usually just normal existing in the environment and just happens to make use of that. And we call that an opportunistic infection. Takes control of that opportunity. We have community acquired fungal infections. This would be the case with things like athlete's foot, very easily transmissible parasitic fungal infection. We have fungal infections that are associated with being kept in a hospital environment. And then, of course, if you have a weakened immune system, you're going to be more susceptible to fungal infections. During the COVID pandemic, it talks about there's an outbreak of mucormycosis. So mycosis is a fungal infection. And this particular kind, mucormycosis, is pretty nasty and it can be disfiguring. It's bad. And they had more than 300,000 people die of mucormycosis during the COVID epidemic because they were more susceptible to this infection from being sick with COVID. And so you usually don't see a lot of outbreaks of that. Moving on to the Protista kingdom. So we have the kingdom fungi, and now we're on to the kingdom Protista. Protista has two subkingdoms, algae and protozoa. Anything that falls into Protista, they can be unicellular or multicellular, but they don't have true tissues. So we're not having distinguishing tissues like we have liver tissue and lung tissue and all these. It's mostly just one multicellular tissue that works together. An example of a multicellular algae would be kelp. Okay. All of the protozoa are unicellular. And you can have single cell algae as well. All right. So the algae, again, these are photosynthetic organisms. They have cellulose cell walls. And these are guys that are primary contributors to our oxygen in our atmosphere now. As far as the eukaryotes go, they actually contribute more than plants do. And they actually can be part of that plankton community, which is a huge food source for a lot of our marine life. But we won't really be talking about algae much in this course. We will be talking about the protozoa. Again, all protozoa are unicellular. And we usually divide them up based on how they get around. So there's 4 ways that you can get around or not as a protozoa. And we'll talk about how they're divided up in that manner later. But they are just single cellular. But their organelles, like their ability to phagocytose things, so eat things up, their ability to move things around in the cell and break things down, they've actually organized their organelles in a way that they look like mouths and digestive tracts and things like that. And they function that way for these unicellular organisms. Very interesting. You really do see that evolutionary bridge going on in these guys. These guys are all heterotrophic, so they get their food from a variety of sources. They don't get it from the sun. So these are either free living, so they just live in their environment and feed on bacteria and stuff in their environment, or they are parasitic. They're parasitic, they're living off of living organisms, right, and causing harm. And that's what a parasite is. Anything that lives off of another living organism and thereby causes harm or damage to it. So there are 3 styles of locomotion, and this is how we're actually going to divide these guys up. You either are pseudopod having, so if you have pseudopods, these are false feet. This is what we typically think of with amoebas. You squish out part of your cell, stick it down, and then just scoot the rest of your cell along with it. Okay, so these are pseudopods, or fake feet, and that type of motion is called amoeboid motion. You can also use that to sort of scoop out and then surround prey and eat it up, like a lot like phagocytosis. Okay, so they can use that amoeboid motion to eat things up. So if you are able to do that, then you would be an amoeba. Or you might have flagella. We talked about those big whip-like structures. Or you might have cilia, those little hairs that beat like oars. We can use those for feeding as well as for locomotion if we are single-celled, which all of our protozoa are. So the 4th group, I told you there's 4 groups. The 4th group are the guys that don't move, are non-motile guys. So obviously these are our amoebas. This is a guy that has flagella, and this is a guy that has cilia. All right. So this guy at the end with the X is non-motile. All of the guys that are non-motile, all of them are parasites. Right? So these guys can exist in two different forms, just like our bacillus and clostridium can. So if you aren't picking up on that, we're heading into similarities with endospores. Okay? So we don't make endospores when we are protozoa. We get different terms because fancy in science. Okay. So trophozoite, these are pretty similar to our vegetative bacteria. And again, one way that I remember this is that troph. So when I think of troph, I think of trough and I think of eating. If you're eating, you're metabolically active, right? You're active, you're existing, you're growing, you're doing things. So that's how I remember the difference here. And that's what our vegetative bacteria do as well. So these are analogous to the vegetative guys. Our cysts are analogous to the endospores. And again, just like the endospores, I promise I can do this, endospores, these are dormant and resting. They're completely inert. So when you get an infection with these guys, just like our endospore forming bacteria, the endospore would get into your body or the cyst will get into your body. But is that what's causing the disease? No. When they become vegetative or they become trophozoite, that active form is what's going to be eating, it's what's going to be causing damage, creating toxins. All that stuff is done by our active form. That's always going to be our trophozoite or our vegetative. And you better believe there's going to be a question on the test like that, right? We actually have one of our critical thinking questions that's kind of like that, or at least it might be in our review week, our weekly review. But anyways, if something is doing something to your body, it has to be the trophozoite for the protozoa. It has to be the vegetative for the bacteria. Otherwise, it's just dormant. It's not doing anything when it's dormant. It's not moving, nothing, right? Cyst is the dormant one. They are also very resistant, just like our endospores are. All right. So again, you can see that we're starting to have characteristics like whether it has flagella or it's non-motile, whether it is, has a trophozoite or a cyst, or what those trophozoites or cysts look like that we can use for identifying factors of these guys. Some pathogens that are associated with these are going to be in this table down here. So anytime that you're studying our protozoa or our helminths, which we're just going to talk about briefly in just a moment, which are parasitic worms, okay, that's parasitology. So a parasitologist studies diseases caused by protozoa and helminths, right? So some examples of our amoebas would be entamoeba. Look, it's in the name. Entamoeba histolytica. This guy causes dysentery. We have Naegleria fowleri and acanthamoeba down here. These guys are our brain-eating amoebas. Very, very uncommon to get infections with these. We actually probably only had like maybe 100 since like 1933. So like in the past like 80, 90 years, we've only had like 300 infections or 400 infections total. Pretty uncommon to get infections with these guys. But if they get high enough in your sinus cavity, close enough to your brain, and they're able to adhere, again, hard for it to do. But if it does, and it gets into your brain, there's almost nothing we can do for you. It's 97% mortality rate, and if you do survive it, you're likely to have lasting damage from these. So it's pretty serious. These guys can live in your hot water tanks. So do not use a neti pot without boiling your water first, please. Okay. Sterilize your water if you use neti pots or use bottled water. Okay. If you use contact lenses, and we'll talk about this in our eye diseases later on. If you use contact lenses, these guys, again, in your hot water heater, if you're not actually using soap to wash your hands beforehand, they can get on your hands and put these in your eye, and this can lead to blindness. Please be careful about that. Sterilize your lenses, make sure you're treating them. Don't just store them in water. Make sure you're using saline. Uncommon, but you've probably seen on TikTok or Instagram or something like that, some videos of people with patches on their eyes. All I do is wear contacts and whatever. Be sure you're keeping your hands clean. Acanthamoeba leads to keratitis. You can become blind from that. All right. Balantidium coli, diarrhea. That is a ciliated. I want to be clear about that. Go back. Ciliated, Balantidium coli. I'm not going to test you over these individual guys. These are just examples, just so you know. Okay. Flagellated, you may have heard of giardia, giardiasis. This is going to be traveler's diarrhea. So if you're going camping or something, even if the stream is bubbling and nice and pretty and clear, it could still have giardia in it. This leads to like 2-month-long diarrhea with malodorous and greasy stools. That's how it's described. Okay, don't do it. Trichomonas vaginalis, a very common sexually transmitted flagellated protozoan. Trypanosoma, these guys are transmitted by a vector-borne. This means that it's transmitted by insect bites typically. So the flying insects that transmit these guys, we'll talk about the diseases later. Leishmania was a common one that we were seeing in our military that were going to the Middle East, getting bitten by sand flies. It's a skin infection. And then our non-motile. So remember, all of our non-motile are protozoan. These are just a few of them. All of our non-motile protozoas are parasitic. These are just a few of them. All right, Plasmodium species. These guys are cause malaria, transmitted by mosquitoes. Very severe and common in sub-Saharan Africa and tropical areas of the world. Toxoplasma. Toxoplasmosis is something that we have in America, as is Cryptosporidium. Toxoplasmosis is maintained within cats. They transmit it through their feces. You clean up your cat litter, and suddenly you have toxoplasma. We've probably, most of us, been exposed to it at some point in our lives. But if you are pregnant, there is like a 1 in 3 chance that it can get spread to your fetus, and it's very high likelihood for stillbirth and loss of the fetus and things like that. So when we tell you, do not handle your cat litter when you are pregnant, we are not freaking kidding. Don't do it. Get somebody else to clean up your cat litter. Wear gloves and a mask when you handle your cat litter, okay? Be careful with it. Then we have Cryptosporidium. This is diarrhea, and it's very resistant to chlorine, so the cysts are easily transmitted. Anybody in here ever been a lifeguard? Ever? Yeah? You've seen people pooping in pools? All the time, right? All the time. Easily transmitted in public swimming areas, okay? Cyclospora, again, diarrhea. All right, moving on very briefly to our Animalia groups. This is Kingdom Animalia. Lucky our lab is going to be short today. We'll get done early for sure today, okay? But just finishing up with our animals, these are our parasitic worms, all right? So there's 2 groups of these guys, the flatworms and the roundworms. The flatworms have another name. It's called platyhelminthes. So I'm going to write platy up here and just follow it with helminths with the E before the S. Platy, I can do it. Platy, okay. Platyhelminthes. And the way that I remember that is if it's platyhelminthes for flatworms and roundworms are nematodes. Platy sounds like flat to me. That's how I remember which one's which, okay? Now for the other guys, I have ways that I remember that aren't going to work for you. They're based on personal experience, but tapeworms are our cestodes, and then flukes are our trematodes. So obviously this is a tapeworm, right? We see this here, and this here is a fluke. Let me put F for fluke, okay? So those are examples of those. We don't have really a good picture on this for the roundworms, but believe me when I tell you we're going to talk about them. Things like heartworms, that dogs can get, hookworms and roundworms that the dogs can get. We get hear this a lot for vets, why I'm bringing up dogs because I have dogs, but cats can get them too, other animals can get them. Those are typically roundworms that we're talking about there. They're also susceptible to our flatworms, our tapeworms, and things like that. And of course, there's human diseases related to these. We don't see a ton of these in America, but believe me when I tell you that they are rampant in our third world or underdeveloped countries. Rampant, okay? When we talk about how common we think a tapeworm is, or even those tiny little, what are they called? The vermicelli-like ones, the pinworms, the butthole worms, right? We talk about those and how common those can be here in the United States. Nothing holds a candle to the giant intestinal roundworm, Ascaris. Ascaris lumbricoides is the most common of the all of our helminths in the whole world. And we don't have that here, but they're so common in underdeveloped third world countries. These are actually a huge issue for them. All right, these are all multicellular organisms, and you can see them with the naked eye. You can, the adults. But we usually don't see the adults with our naked eye because they're usually in the body when they're adults. So we will see their larvae or their eggs, and those will typically be microscopic. You usually get these guys from eating food that's contaminated, drinking water that's contaminated, or they can even get through unblemished skin. They can get in through your freaking intact skin. They can get in through your skin, you guys. So they can, like, not all of them, but there are certain species. This will help you sleep at night. There are certain species that can literally like live in water that's been contaminated. They usually have a life cycle that involves snails, and they'll like come out of the snails and get into your skin, like just swimming in the water. It's called swimmer's itch, and it's really common in tropical areas, okay? And then there's some of them that can actually get in through the soles of your feet. You think that your feet being thick skin, that you'd be protected, but you wouldn't be. So these, again, are more common in our sub-Saharan Africa and tropical regions of the world, but they can get in through intact skin, and all that. So the host where the actual reproducing, where the egg producing and things like that happens, that's called the definitive host. We'll talk about these guys when we get to chapter 23. We'll have examples and talk about the diseases they cause. We're going to talk about how you get them. Usually it's going to be through oral ingestion and contact with soil and things that have been contaminated like that, or contact with water. But insects can also act as way to transmit these guys. These are a little easier to kind of look at macroscopically as well as in the microscope, seeing the eggs and the larvae. They're usually pretty easy to tell the difference visually. And here's our little breakdown of these guys. Our kingdom Animalia. So we have the helminths. This is our platyhelminthes. These are our flatworms. These will be broken down into our cestodes and our trematodes, whereas we just have roundworms for the roundworms. They are going to be nematodes, and they can be either maturing in humans or not. If they don't mature in humans, that doesn't mean they don't cause disease in humans. It just means they don't have a definitive host as humans. Okay. So yeah, that's that. Our questions really quick. The biggest difference between eukaryotic cells and bacterial cells, this is the presence of a nucleus, a true membrane-bound nucleus for our eukaryotic cells. What is the nucleus for? It contains our genetic information, our DNA, the control center of the cell. What is the function of the mitochondria, the energy producer for the cell? Subkingdoms of Protista. These were our protozoa and our algae. And then the last question, what is the scientific term for flatworms? Platyhelminthes. I did not make William have to say that. So platyhelminthes. So that's it. So we'll go up to the lab now. Then we'll give you guys a little bit to recoup and come in and get signed in for our Kahoot up there. And we'll get started right away. Again, we'll be done early today for sure.